Hi everyone. I'm going to start this presentation on photosynthesis with the equation for photosynthesis shown here on the screen. Remember that photosynthesis involves an input of solar energy, carbon dioxide from the atmosphere, water, and photosynthesis converts those reactants into glucose and oxygen. As we go through the presentation, keep in mind the connection between photosynthesis and the process of cellular respiration that we've already discussed. Just a reminder about the anatomy of a leaf. The important components to leaf structure facilitate how photosynthesis works. So remember, we have the stomata or stoma, which is singular, which is the opening in the leaf required for gas exchange. There are always more stomata on the underside of the leaf than on the top of the leaf to prevent water loss. In addition, the air spaces in the spongy mesophyll layer allow the air to access all of the cells, all of the mesophyll cells that are going to be the sites of photosynthesis. Furthermore, the upper and lower epidermis provide structural support. Notice the lack of chloroplasts in those cells because their primary function is to secrete the waxy cuticle, which is the water impermeable layer that is always thicker on the top of the leaf to prevent water loss. In addition to the structure of the leaf, understanding the structure of a chloroplast is also very important. So a chloroplast has a double membrane just like the mitochondria does, and inside of the chloroplast, the filling inside is called the stroma, and then we have the thylakoid stacks, which are continuous membrane discs that are the site of the light reactions. In the past, scientists always theorized as to where the oxygen gas came from that was released as a product of photosynthesis. Initial theories were that oxygen gas came from the carbon dioxide that was invested in photosynthesis, when in fact the oxygen gas that is produced as a product of photosynthesis actually comes from the water. So we know this now that we understand that the splitting of water is a critical component to the light reactions. So the oxygen in carbon dioxide is actually a component of the sugar molecule and the water that is also a product of photosynthesis. Understanding what is oxidized and what is reduced is very important to understanding the process. So carbon dioxide is reduced, meaning it gains electrons throughout the series of reactions, leading to the formation of glucose. And the water molecules are oxidized, which means they're losing electrons in the process of being split in the light reactions and becoming the oxidized version, which is oxygen. This here is simply showing how the water molecules that are utilized during photosynthesis are going to, these water molecules, the electrons from those are ultimately going to be invested in the carbon dioxide. So the water is oxidized and the carbon dioxide is reduced. In terms of where the different reactions occur, the light reactions are what occur in the thylakoid membrane. And remember, they involve the input of photons of light, water, and through those two reactants, we get a production of oxygen gas and the production of ATP and the electron carrier NADPH. We will break down the light reactions in a little bit more detail now. But first, understanding that the products of the light reactions actually power the Kelvin cycle. So ATP and NADPH, which are the important products of the light reactions, are invested in the Kelvin cycle along with carbon dioxide. This occurs in the stroma of the chloroplast and recycles then the NADP plus molecule and the ADP molecule back in. Through the process of the Kelvin cycle, sugar G3P is produced, which will lead to the formation of glucose. All right, now to break down more specifically what happens in the light reactions. In the thylakoid membrane are photosystems, and as we discussed in class, photosystems are responsible for harvesting light and using that light energy to power redox reactions. So the photons of light hit the pigment molecules located in these light harvesting complexes, and those pigment molecules have electrons that are excited when the photons of light hit them up to a higher energy level, and when they fall back down to their resting state, it releases potential energy, which then triggers the next pigment molecule to have its electrons energized to a higher energy level, and when they fall back down, energy is released again, and that energy triggers the next pigment molecule to have its electrons excited to a higher energy level, and they fall back down, and that energy continues to be passed along until it is funneled to the special chlorophyll molecules located in the reaction center. Those chlorophyll molecules are actually transferred to the primary electron acceptor. So once those electrons are excited, they are actually involved in a redox reaction where they are donated to the primary electron acceptor, and then the primary electron acceptor is reduced and will donate its electrons to the electron transport chain that we'll talk about here in a moment. Again, these photosystems, this is a general photosystem, but two photosystems and an electron transport chain are what make up the light reactions in the thylakoid membrane. Let's take a closer look at what the light reactions actually involve. So, like I said before, there are two photosystems that make up the light reactions, along with an electron transport chain between them and a small electron transport chain that comes after the second of the two photosystems. So the first photosystem is actually titled photosystem 2 because they are labeled in the order in which they were discovered. So again, when light hits the first photosystem, that cascade of energy occurs until the energy reaches the special chlorophyll A molecules. At this time, those chlorophyll molecules, due to that investment of energy, they are oxidized and the electrons are donated to the primary electron acceptor, which is in turn reduced. The electrons from the primary electron acceptor then begin the journey down an electron transport chain in the thylakoid membrane. This electron transport chain is very similar to the electron transport chain that we looked at in cellular respiration. There are a series of proteins that increase in electronegativity as the chain progresses, and as those electrons move to higher and higher electronegative molecules, that generates a release of potential energy, which drives a proton pump here in the cytochrome complex. The hydrogen atoms that are in the stroma of the chloroplast are pumped into the thylakoid lumen. We will take a look at an image that illustrates that directionality of flow on the next slide. While those electrons are moving through the electron transport chain, this void of electrons in the special chlorophyll molecules needs to be filled, and that's where the splitting of water occurs. So a water molecule is split by a water splitting enzyme that is the source of the electrons to fill that void of the electrons that have been donated, and that's also where the oxygen gas is produced from that splitting of water, and the H plus ions simply add to the concentration of H plus in the thylakoid lumen. Going back to the electron transport chain, as electrons are moving down the chain, the second of the two photosystems, which is actually photosystem 1, is also absorbing photons of light and funneling the energy from that light through the pigment molecules ultimately to the special chlorophyll molecules in this reaction center. Those electrons behave the same way in that once they are excited, they are removed from the special chlorophyll molecules, so they are oxidized, and the primary electron acceptor is reduced. The electrons from this photosystem continue down a very small electron transport chain involving a transport protein called ferredoxin, which passes the electrons to an enzyme embedded in the membrane called NADP plus reductase. The electrons are then utilized by this enzyme along with NADP plus to reduce NADP plus to NADPH, which is the electron transporter that will play a role in the dark reactions. Because a void was created by the donation of these electrons, that is filled by the electrons coming down the electron transport chain. So as I said before, this electron transport chain is very significant because it will lead to the production of ATP, which we can see a little bit easier on the next slide. So as you can see here, this gives you a better spatial perception of what the photosystems and the electron transport chain's positions are relative to each other. Here is photosystem 2. Here's the electron transport chain with the cytochrome complex hydrogen ion pump. Here's photosystem 1 with ferredoxin and the NADP plus reductase. Down here is ATP synthase that's going to be the site of actual generation of ATP. So as I was saying before, as the electrons are moving through this electron transport chain, the potential energy that's released will power the cytochrome complex to pump hydrogens from the stroma, where there's a low concentration of hydrogen, into the thylakoid lumen, where there is a higher concentration of H+. So the H++ is coming in from the cytochrome complex, and the H+ is left over from the splitting of water will fill the thylakoid lumen and passively diffuse through ATP synthase, spinning it and opening up the active sites for ADP and inorganic phosphate to bind. That then will generate the ATP. Remember, this is passive transport, and the transport going on here in the electron transport chain is active transport because the hydrogens are moving from low to high concentration. Now we have built ATP, and now we have built NADPH, and now the Calvin cycle is ready to begin. However, before we get into the dark reactions, I do want to take a moment for a little aside of something that your book mentions, referring to a time when a cell might be in crisis, when there is not enough ATP present to continue on to the Calvin cycle. And this is what we call cyclic electron flow. So the previous slide involves or is described as non-cyclic electron flow because the electrons are moving in a linear path from photosystem to photosystem and ultimately to NADPH. However, if more ATP is needed to be generated, the chloroplast could sacrifice the usual pathway from the photosystem 1 to ferredoxin and ultimately to NADP plus reductase to make NADPH, and instead that ferredoxin can pass those electrons back to the cytochrome complex. Doing that would again generate energy to power the proton pump and ultimately put more hydrogen ions in the lumen of the thylakoid to flow through ATP synthase to make ATP. Chloroplasts and plant cells will only do this though if there is a shortage of ATP that needs to be addressed, because in doing this, the cell is not making NADPH, and so the Calvin cycle will not happen without that NADPH. So this is just done at a time when ATP might be short, and then the chloroplast will have to kick back into non-cyclic electron flow in order to reestablish the production of NADPH. Once ATP and NADPH are in proper concentration, the Calvin cycle can begin. So the Calvin cycle, remember, happens in the stroma of the chloroplast, and this is where we see the investment of carbon dioxide from the atmosphere. The important components to the beginning of the Calvin cycle, called the carbon fixation phase, is the special enzyme rubisco. Rubisco is the enzyme that actually fixes carbon dioxide to the initial intermediate called RUBP. Carbon dioxide and RUBP will be joined together by rubisco to form the first intermediate of the process, and that intermediate will continue to be processed as the second phase of the Calvin cycle occurs, which is called the reduction phase. So with the investment of 6 ATPs and 6 NADPHs, these intermediates will be continuously reduced into the product of G3P. Now pay careful attention to the fact that G3P, there are 6 molecules of G3P that are produced, and only one of those G3P molecules will actually be outputted from the Calvin cycle and used to eventually make glucose by the cell. The rest of the G3Ps, the other 5, are going to be used to regenerate the RUBP. With the addition of even more ATP, the RUBP is going to be regenerated through a series of intermediate steps, and that RUBP is going to be needed for the next round of the Calvin cycle. Some of the important observations here to make is that there is a large energy investment and a large electron investment to this process for a very small output of G3P. So this is a very energy taxing process. This is simply a schematic of the relationship between the process of photosynthesis and the process of cellular respiration that we've already talked about. This kind of interconnects the reactants and products of both of those reactions or both of those processes, and should be something that you can explain and even add more detail to in your explanation in order to integrate these two processes together. Lastly, I want to end by talking about the special case of photorespiration, which really is a negative situation that a plant might find itself in when it is trying to prevent water loss. So at very high temperatures, when water is scarce, plants typically close their stomata in order to prevent wasteful water loss in the leaves. But in closing the stomata, you are now, as a plant, preventing gas exchange. So as photosynthesis continues to occur, oxygen levels begin to build up in the leaf, causing the oxygen concentration to be very high. Whereas CO2 levels are going to continue to get lower and lower because there's no way for carbon dioxide to enter the leaf if the stomata are closed. So the plant is attempting to conserve water, but creating a gas imbalance at the same time. Another negative repercussion from closing the stomata is that rubisco, the enzyme involved in the Calvin cycle, has an affinity for both carbon dioxide and oxygen. When rubisco binds CO2, the Calvin cycle occurs. But when CO2 levels are very low, oxygen will more often interact with the rubisco, and in doing so will lead to additional problems. When rubisco adds to the Calvin cycle, glucose is not produced. In fact, a 2-carbon product is produced that does not provide the cell any useful inputs into cellular respiration like glucose does. In fact, it does not provide the cell any um, energy uses and instead simply causes there to be a lack of sugar and therefore a lack of reactants for cellular respiration. And therefore, in the long run, no ATP would be able to be produced in cellular respiration since glucose is not being made. A plant cell would simply break down this intermediate product that is produced, and the peroxisomes would play a role in that. But this would be very critical for the cell because if it's not making glucose, it will not be able to survive for very long. Plants that live in really dry places, therefore, have had to evolve to come up with a way to avoid this photorespiration and this production of wasteful products when oxygen levels are high. Therefore, I'm going to talk about two different types of plants that have created modifications to the process so that they do not photorespirate and instead can overcome that and still efficiently produce glucose. So as you can see here, I'm just removing some of these notes from when I presented this in class. So basically, the first plant I'm going to talk about is called C4, and C4 plants have spatially separated the process of the Calvin cycle from an initial process that they do in order to fix carbon dioxide. So there are a couple significant differences between a C4 plant like this and the typical C3 plants that we've been talking about. So C4 plants have special leaf anatomy. So as you can see here in the picture, there are the spongy mesophyll cells, followed by the more structured palisade cells. But then below those palisade mesophyll cells, there's an additional type of cell that all plants don't have, which is called these bundle sheath cells. As you can see with how they are layered, the bundle sheath cells do not have access to the air pockets found in the other parts of the leaf. This is significant because in these bundle sheath cells is where the Calvin cycle is going to happen, and in these mesophyll cells is where this new process is going to occur. So let's take a look at what that new process is. Here's an example of a mesophyll cell. Something important to note is that there is an additional enzyme that is very unique to C4 plants called PEP carboxylase. PEP carboxylase is unique because it can only bind carbon dioxide. So think about this. If the stomata are closed and carbon dioxide levels are very low and oxygen levels are very high, the plant is basically ensuring that carbon dioxide will be the gas that is initially fixed into the process because oxygen gas cannot interact with PEP carboxylase. And also remember, the mesophyll cells are what are nearest to those air pockets, and so having an enzyme that only binds CO2 is a huge advantage. Once the CO2 is fixed to this intermediate called PEP, a 4-carbon product is produced, and that 4-carbon product is continuously processed and ultimately funneled through these plasmodesmata into the bundle sheath cells down below. In the bundle sheath cells, the intermediate is broken apart into carbon dioxide and pyruvate. The significance of this is the bundle sheath cells contain the Calvin cycle, and so at this time, carbon dioxide is the only gas that Rubisco will have access to, and the Calvin cycle will proceed as normal. And so glucose will be produced and sent into the vascular tissue for transport. The pyruvate that is produced as the intermediate molecule is broken will be recycled back into the mesophyll cell, and with some ATP will be converted back into PEP, so the process can repeat. So again, there's a spatial separation between the fixation of carbon and the Calvin cycle itself, ensuring that Rubisco is only getting carbon dioxide. The other modification that some plants have made is instead of a spatial separation of carbon fixation, it's a time of day separation of carbon fixation. These plants are called CAM plants. So CAM plants contain PEP carboxylase just like the C4 plants do, except they're going to fix that carbon dioxide into the 4-carbon intermediates during the nighttime when they can safely open their stomata and prevent water and minimize water loss because it is nighttime and the temperature is cooler, and they can open their stomata and let in carbon dioxide. That carbon dioxide will be fixed into a 4-carbon intermediate, which the plant will then simply store until the daytime. In the daytime is when the stomata are closed in order to prevent that water loss during the hottest and brightest part of the day. And so during the daytime, the plant will break apart that 4-carbon intermediate and release carbon dioxide into the Calvin cycle. So the Calvin cycle will happen during the day when the stomata are closed, but will still have a bountiful access of carbon dioxide because of the breaking down of those 4-carbon intermediates to provide the Calvin cycle the carbon dioxide it needs so that sugar molecules can be produced. So just to recap, CAM plants have temporal separation of their carbon fixation, initial carbon fixation, and the Calvin cycle, whereas C4 plants have spatial separation of the initial carbon fixation and the Calvin cycle. Both of these are adaptations that these plants have made to hot, dry, arid climates in order to prevent photorespiration from happening.